paraquat, if you administer that in this animal model, it takes a little bit longer to develop, but it still turns out to be the similar situation. So back to this, so again, with paraquat, MPTP, or some genetic component, or just because these neurons are so sensitive, so that's, that these neurons start to get destroyed. But again, I mean, probably in all of us, those neurons are getting damaged as just we live our lives. So, but you need upwards of 75% cell loss in this little particular region to develop Parkinson's-like symptoms. So, in some cases, it's good because we do need to get to that level of damage, and we are somewhat safe of, most of us will not develop Parkinson's disease. But it is a sensitive part of the brain, high rate of metabolism, and this iron, and then you have all these other factors, bombarding that part of the brain. Oops. So this is what it would look like in a human. This is a bit better of a picture. This is what it would look like in a human brain, in those midbrain dopamine neurons in an individual with Parkinson's versus a normal individual or normal substantia nigra. So again, you can see the difference. So this would be, it's probably not an experimental model. It's probably not that this person was given high doses of paraquat or MPTP or any other drug, but they developed Parkinson's. And then on autopsy, if you look at their brain, you can see in the normal brain all these brown dots that are indicative of those dopamine-producing neurons. And in the Parkinson's brain, they're pretty much gone. And so that, when you have that, you have a reduction in the release of dopamine up to this part of the brain, up to that, so this is now a human brain as well, up to that basal ganglia. That's really important for organizing our muscle-driven motor movements, as well as our thoughts that go through here. So there's this part of the brain gets to be really, really complicated, and my students really, I've been able to work through it with my students, but to describe how it's working, there's a lot of inhibition, excitation of inhibition, inhibition of inhibition, that will help us essentially initiate a movement or stop a movement. So if you have any imbalance in the chemistry of this part of the brain, then movements are affected as well as thoughts. So when I talk about this, I talk about caffeine, because that's the drug that I'm most familiar with. Caffeine actually affects this part of the brain. So what it does is it impairs your ability to inhibit particular movements. So you start, you get jittery, you start tapping your hands, start moving around a lot when you're on caffeine. But it also impairs your ability to stop thinking about things. So you get into that rumination set of those phases. So you have these circles that are just thoughts are just going around and around and around and around, and you start tapping your hands, and it's really, so it's a part of the brain that it really explains a lot of our behaviors, and a lot of disorders are actually based on interruptions of the function of that part of the brain. Huntington's disease, obsessive compulsive disorder, anxiety disorders, Parkinson's disease that we're talking about now. So it's a really, again, it's a fascinating part of the brain, and but when you lose dopamine here, in the case of Parkinson's, that's the part where then you get the shakes, like you could have when you drink lots of coffee, or if you have Huntington's disease. But there's also a separate pathway that goes through there that disrupts your ability to initiate particular movements. Yep.
Is substantia nigra bilateralized, or is it concentrated in a single location on the midbrain?
Yeah, so it's actually on both sides. Yeah, so it is bilateral. Yeah.
So why would it seems to me that once you impair that system, it affects everything. Why does not bilateralization not provide some redundancy and protection?
I guess essentially both sides are getting damaged at the same time. Right? So both parts, so even though it's on both sides, it's not that only one side gets damaged, it's both parts that are getting damaged. And so when you lose, dopamine in this case is working to, I guess what we'd say is modulate the movement. So it's not like you can still move clearly in Parkinson's disease, but the movements aren't as fine as they should be. So it's like the language abilities, you can still talk, but they're just more choppy. So dopamine is kind of, sewing it all together and making things nice and smooth. Yeah.
You mentioned caffeine. Does it have a permanent effect in the long term, you think?
That is a good question. So we go back to nicotine in this case as well. So nicotine and caffeine there's evidence that they're protective against developing Parkinson's disease. So, and again, that's not really all that clear how that works. So yeah, nicotine, the magic drug, can do everything. But so yeah, it's hard to say whether caffeine has permanent effects in that part of the brain that are detrimental, but at least some people have said that it's actually protective against Parkinson's. So keep drinking your coffee. 1103. Okay, so this would be what a Parkinson's rat brain would look like anyway. So when you have the cell loss down here, and you're looking at how much dopamine is being produced up here, what you can pretty clearly see is that all this sort of dark maroon red is what is indicative of dopamine. So this is the intact side. So this is where now, if they've made a lesion down here in the substantia nigra only of one side, I don't know if this might address your question. If you only lesion one side, if you lesion one side, this side is intact, you can see that there's all this dopamine here. But if the substantia nigra is destroyed on this side, then there's no dopamine output there. So in Parkinson's, it's actually both sides that are being damaged. Yeah.
So I would infer from that in that case, there would be no apparent consequences of the damage to one side.
Yeah, well, right. So in fact, in these rats, oftentimes you see only impaired ability on one side of the body. Yeah.
Also, the dopamine, sorry to belabor it, but so the dopamine being produced by one side of the substantia nigra, it only services that hemisphere of the brain.
Yeah, exactly.
So
Yeah.
it would localize the consequences of the
That's right.
on the other.
Yeah, exactly. Yes.
Just a quick question. Can an MRI see this, or do you only see this in autopsy?
No, well, yeah, so because it's such a small part of the brain, it's really hard to detect that damage. And again, there's a lot of different cell types. I should have probably, I don't know if we can see it on this one. I guess kind of. There's a lot of different, maybe you can't see that, but there are these lighter purple cells down here that are part of that substantia nigra that are fine. It's just those, these darker dopamine producing neurons that are the ones getting destroyed. But I'm trying to think of what the percentage of dopamine neurons is in that substantia nigra, but let's say it's only about maybe 50% of the neurons in that part of the brain are dopamine producing. So to see that level of damage, I don't think you could do that with an MRI.
Any genetic component?
Yeah, I'll try to touch on that a little bit. I have some pretty complicated slides that show all the factors that go into the development of Parkinson's. So the basal ganglia is, it's not the site most affected, it's actually the output site that's most affected. So the site most affected are those cell bodies in the base of your brain, and then the projections, the axons, the dopamine that gets dumped out is up in the basal ganglia, and when you lose that dopamine up there, that's where you see these symptoms. So inability to control not only your movements, but also thoughts, emotions. So again, thinking about caffeine, if I have a coffee at 3 o'clock in the afternoon, I'm not sleeping that night. So because I'm sitting there in bed, I'm not, shaking or anything, but I can't stop thinking about things and think about what's happening in the day. And, it's just like, okay, stop. And I just can't do it. So with caffeine that happens, but in Parkinson's, that happens quite a bit. And you can see too how this is related to obsessive compulsive disorder, because you can't stop thinking about things, and as well, then you can't stop, you can't inhibit your movements, your behaviors to act out those things. So the basal ganglia assists back to the frontal lobe. So, the frontal lobe, yes, is the CEO of the brain, and it's the executive, it does a lot of things, but it has a really strong supporting cast. And one of those supporting casts are the basal ganglia. So it's helping the frontal lobe sort of, as it says, prioritize transfer of information, prioritize things that are important for your life, paying attention. So again, you think about in attention deficit hyperactivity disorder, the basal ganglia is thought to be disrupted in that case. In that case, they think there's high levels, I don't want to say high levels of dopamine, but changes in the dopamine tone so that the basal ganglia can't help the frontal lobes keep things organized in working memory and help you pay attention. Okay, so I don't need to get into that. So this is just kind of an overview, and again, the level of detail that's shown here is not required for any of this, but it's, to say again how fragile those dopamine neurons are. So this would be a dopamine producing neuron right here, and you can see that the toxin, agricultural toxins, so the paraquat, can actually get in and set off this cascade of events that will cause damage to that neuron. You have these drugs, MPTP, that can also enter through the blood-brain barrier and get into those dopamine-producing neurons and set off all these cascades that will ultimately end up destroying those neurons. So again, not only are these neurons really working hard, I mean, these dopamine neurons are really functioning at a high level in all of us to, again, to keep everything organized, to keep everything moving smoothly, to help me so that I can, if I need to, I mean, this is one thing that I've learned from my videos is I move my hands a lot. So if I can say now, okay, now my dopamine system has to get moving to stop me from moving my hands, which is really difficult. But so as I'm doing that, as I'm trying to stop these movements, my dopamine neurons are working overtime, and they're producing all these, again, we go back to these reactive oxygen species, these toxins that are just being generated. Typically your brain takes care of those, but unfortunately in these dopamine neurons, it's such a high level that the brain can't keep up, so you just lose these neurons in general. But like I said, you need to lose about 75% to really get full-blown Parkinson's. So you can imagine that high rate of activity here, plus these toxins or plus these drugs, can all sort of start to mish together and really cause some damage, long-term damage to those dopamine-producing neurons. And then it gets even more complicated. So this is here's your genetic, so there's a genetic contribution, so mutations in the PARC2, so Parkinson's 2 and Parkinson's 3 genes. There's a Parkinson 1 gene. There's all this idea about misfolded proteins, so the toxins coming in, producing this mitochondrial dysfunction, that's what I showed on the previous slide. Oxidative stress, so those reactive oxygen species, the low level of antioxidants, the high iron content, all just converging on all these different pathways and then ultimately leading to neurodegeneration. So, and then the unknown mutations in synaptic vesicle proteins. So there's a lot of unknowns in this whole pathway or this whole convergence. And like I said, it does get to be a bit of a mess, but it's also, obviously leading to new treatments and trying to understand how we can actually stop the progression, stop this, the bottom line, how do we stop that neurodegeneration to reverse those motor deficits?
Does exercise help increase the level of dopamine?
So does exercise help increase the level of dopamine? I think so, as long as you find it rewarding. So as long as you're not being forced into it. So I guess if you put a running wheel into a mouse cage, they actually go in and they'll do it. They enjoy doing it. But if you force them to do it, then it's less positive. So I mean, so again, in terms of just in general in dopamine, the more rewarding something is, the more dopamine is being produced. So or the other way around, the more dopamine there is, the more rewarding something is. So if you are engaging in something that's pleasurable, then dopamine levels will increase. And if, walking, exercise, they do have pleasurable effects, so that can increase dopamine. And then it gets to be even worse. So again, I'm not even going to try to talk our way through this one, but I think, thinking about some of the disorders that we've been talking about, so MS, we'll see this in Alzheimer's as well, what are called the microglia, so the inflammatory component of all these diseases or these disorders. So there's these inflammatory systems or these inflammatory neurons getting into the brain and then dumping out all these toxins that, I mean, there's these microglia are meant to clean up junk really in the brain. So in Parkinson's, they're meant to clean up some of these proteins that are getting in the way. And actually this will become much clearer when we talk about Alzheimer's disease. And somebody had sent me a link to a paper about how there's researchers looking at these microglia responses, these inflammatory processes, as a way to stop the progression of Alzheimer's disease, which I think is really, again, I think it's a nice way to, as we close up this course, to think about how these microglia, these neuroinflammatory processes are now being targets for treating different types of disorders, MS, Parkinson's, Alzheimer's, anything that can, where the microglia get overactive and they spit out all these toxins that start to destroy these neurons. So it's this cascade, and again, I'll talk a bit more about it when we get to Alzheimer's, but it's something to keep in mind that I think a relatively common denominator between a lot of brain disorders are these neuroinflammatory processes, are these microglia that get activated. They're meant to clean up stuff, junk in the brain, so in Alzheimer's it's clean up those plaques, but what they ultimately start to do is spit out their own toxins, they make the system work harder, and then more damage can actually happen. So it's kind of a pain. So what are the treatments? So here, behavioral treatments. So again, maybe this physical therapy will help to get some of the dopamine levels back up. Again, as long as it's pleasurable. The most common treatment is what's called Ldopa, so it's a pharmacological agent that's essentially a precursor to dopamine. So because essentially they're short-circuiting this loss of dopamine, they're just artificially replacing dopamine. That increases the levels, and that often will help with the movement disorder that's seen. And there was a movie, Awakenings, back in the 80s, I want to say, with Robert De Niro, I believe. I think he was either the doctor or he had Parkinson's, and that was the treatment that was experimental at the time. They gave him this Ldopa, and he was able to get up and walk across the room, and just remember that scene. So so I mean, there's also some other drugs that are meant to help control some of the other symptoms, but again, you're essentially, with the pharmacological treatments, you're treating the symptoms, so you're not treating the actual underlying pathology that's continuing. Yep.
But I don't understand why that would not be self-sufficient. The only problem is this fancy night on there with produce dopamine, and you would think that you could cure this unconditionally by bathing the brain in dopamine.
Yeah, so you can definitely produce some long-term relief of the symptoms, but again, dopamine by itself now is also toxic. So you're making these, so again, you can't just inject dopamine into the body because it doesn't get into the brain. But this Ldopa, the precursor to dopamine, can actually get into the brain and then be converted into dopamine. But again, high levels of dopamine can also be toxic to the brain. High levels of dopamine are associated with drug addiction. So there's a lot of other changes that can be happening in these output regions. So I don't know if I have a picture of that. So what you're essentially doing is trying to get whatever, so again, that 25% of dopamine producing cells that are left of spitting out their dopamine, you're making those work overtime now by sticking in this Ldopa. So that at these terminals, with that Ldopa, that will produce more dopamine. So, but again, it's those 25% that have to make up for the lost 75%. So then they start working overtime, and it's not a long-term solution.
I'm still confused as to why, you can certainly penetrate the blood-brain barrier by direct insertion. You think that the dopamine mechanism is doing that, and you think that in terms of balancing the relative amount of dopamine, you think that trials will lie. Certainly there might be side effects, but certainly we need the condition more than outweigh
Yeah, well, again, so like it's with any long-term drug actions, is there's going to be some side effect, and there are different, so one example is antidepressants, so they increase serotonin, but oftentimes when people have high levels of serotonin, they start overeating, they start to, in fact, hallucinate, so they have all these other, side effects that potentially could be even more detrimental. So you can imagine that if you're eating high-fat foods, your heart is not going to be functioning so well, and that could lead to death. So in the case of dopamine, it's, all of a sudden you're increasing your reward pathways, and there's ideas about mania and doing things that might not be so good for your long-term health. So and also there are ways now of penetrating this blood-brain barrier, so you could actually get dopamine in, but then again, once you penetrate that blood-brain barrier, then your brain is open to infection, and that's a pretty, there's actually some new evidence that came out this week where if you go through the nose, you can actually get through the blood-brain barrier and effectively deliver drugs. But again, the problem that most neuroscientists have with that is once you penetrate this blood-brain barrier, then you're open to infections. And if you get an infection in the brain, then, you know, lights out. So I think that, let's just get up to this. So the stem cells, I think, are really, are probably, you know, like in the case of a lot of these diseases or disorders, are the next step. So, you know, in this one, it actually makes quite a bit of sense. So, you know, just to review a bit, you have this gradual loss of those dopamine-containing neurons. You have the symptomology, you have the motor difficulties. So the current therapy is replacing dopamine or increasing levels of dopamine, either through that Ldopa or different drugs. So dopamine receptor agonists, some of the best ones are amphetamine, cocaine, heroin. So those will increase dopamine massively in your brain, but that's not a long-term solution to fixing Parkinson's. So again, I think pharmacological agents are one thing, but I think, you know, what probably holds promise is stem cell replacement. So again, if we go back to this picture, if you remember, you have those decreased dopamine inputs to this motor center, this thought organizing center. So what you want to do is, in fact, then get dopamine or get stem cells or some precursor cells into that area so that it turns out to this. But so again, the one thing that I'll make the point again about getting stem cells in is that those cells not only have to now start to produce dopamine or, you know, become dopamine producing neurons, but they have to get that dopamine all the way up here, and then now this part has to recognize that this new dopamine is coming in, and then to go up and start reorganizing the frontal cortex. So it's complicated. So Michael J. Fox is obviously a big proponent of stem cell research, and it's true that what's really, you know, good about this is that scientists have been able to put stem cells into a dish and get them to produce dopamine. So they can actually make dopamine neurons from stem cells from a variety of sources. So like what we were talking about last week with the skin cells, is you can take a skin cell and turn it into a dopamine producing cell in a dish. So what happens then when you put it into the brain is basically unclear. So at least as far as I know, there's been no clear demonstration that this actually works. When you put it into the brain, so if you have these stem cells in a little petri dish, you take them out and you inject them right into that part of the brain, whether or not they're able to, so reinnervate, so get those projections back up into this part of the brain, and then produce dopamine to recover from those deficits from that Parkinson's disease. That's the stumbling block now. And again, thinking about what Michael J. Fox has been arguing, that's why we do really need more research. And I'm not just preaching or anything, but I think it's really true that we're at the point now where it's really, really close. And there's probably, who knows, maybe a year or two, people will have been able to figure out how to actually do this, get it into the brain, and produce dopamine. But again, you have to think about how the brain develops to be able to get those neurons into that part of the brain, get the adult brain to be more like an infant brain so that those cells can actually send up their projections that pretty far distance. So it's nothing, I guess it's easier to see it here. So these little neurons have to get axons through all this other stuff up into this part of the brain to be able to then, spit out dopamine, which is, again, not that easy of a task. Perfect, 11:20. Question?
It's not a very accessible part of the brain, is it?
No, well, that's the other problem, is it, down on the base of the brain. So, I guess you'd have to go through maybe the back here and then, put a little needle all the way through. And plus on the way there, the damage that you might produce to other structures is, and, not to mention the possibility of an infection. So again, it's like, not to belabor this mess, but once you get neurons going in there, you get these stem cells, so you're still going to get this